Hi, friends. And thank you, Chairman, introducing me and chairing the paleo session very nicely. Well, I'm really honored to receive this prestigious award like a paleo award. When I first heard the news of paleo award, I said in mind, oh, thank God. How can I take this honor of becoming an awardee of such a prestigious award? Of course, I was so proud of myself, but I felt a great sense of responsibility. So I thought that this Bailey Award could be accepted as an encouragement of doing more work on clay science before getting older. Well, so I have to thank all the colleagues of Clay Mineral Society, of course, those who recommended me and also nominated me and selected me as an awardee for this Bailey Award. Well, my thanks are also extended to my previous students and postdocs and the other colleagues in Korea and also in the world, those who actually collaborated with me with the passion and endeavor in order to realize the new bioclay hybrid materials. Well, today I really want to share this honor with my former teacher, the late the Professor Armin Weiss, at the Institute of Inorganic Chemistry at the Ludwig Maximilian Universität München. During this period of time, I was a graduate student, PhD student in his laboratory. I stayed there for about 4 years, and as one more year as a postdoc, and then I moved to Oxford. And actually, the Professor Armin Weiss proposed the hypothesis of the origin and the evolution of life from clay. Probably some of you may remember his paper. Without him, I think I would not be here today. Well, thanks to him, I was able to take the first step in clay, clay community by presenting a paper in the 6th International Clay Conference, which was held at Oxford in 1978. It was almost 45 or 44 years before. Yeah. During this period, as I told you, I was a PhD student in his lab, where I learned 2D structural future of clay minerals like kaolinite, halloysite, monazite, elite, etc., etc. And the anisotropic nature of physicochemical properties and the host gas chemistry are based on intercalation, reaction mechanism, etc., etc. Well, if you allow me to tell you a happy memory with my teacher that I cannot forget when I visited the Oxford. And we were hanging around the several, back streets of Oxford in order to find bars or pubs where they can provide the bitter, German beer. Yes, and 3 years later, I participated in the 7th International Clay Conference with my teacher again, which was held at the Bologna, Italy, in 1981, which was also 41 years ago. And I was able to present the 4 papers with my colleagues, Dr. Heinrich Meyer or Dr. Otto Becker. At that time, I was a postdoc fellow at the Inorganic Chemistry Laboratory, Oxford, in England. And my supervisor, Professor appointed as a new director of inorganic chemistry laboratory. He hired me as a postdoc fellow, and I was involved in a team, research team in developing the new semiconductor with a narrow bandgap energy for H2O electrolysis to produce H2 gas as the green energy. But I had to come back to Korea from fourth semester in 1981, since I got a tenure track position at the Seoul National University, so that I cannot continue to work further at the inorganic chemistry laboratory there. As you probably know, that the Professor John Goodenough became the oldest person to receive the Nobel Prize in Chemistry in 2019, 3 years ago. And now he's 100 years old, but he's very energetic. And he, as far as I told, that he comes to the laboratory every day. Yeah. As I told you, I was very lucky to have such a great teacher during my career. And this Professor Goodenough was honored for pioneering breakthroughs that led to to develop the 2D cathode materials and the widespread use of the ion lithium batteries. So I was also learn more about 2D materials and how that kind of 2D materials can be applicable for the clean energy research. After coming back to Seoul National University from Oxford, I became interested in the fundamental research, such as the symmetry, electron structure, property relation of transition metal ions, those which are stabilized in the low dimensional solids, 1D or 2D materials. Well, around 1995, I thought that the materials with a submicron size would be very interesting in terms of physical chemical properties. So I decided to organize the first nano conference in 1996. And I was trying to invite the professors from in the world, those who are working on the molecular clusters. As you know well, nanotechnology became hot potato after an announcement of the nano initiative by the President Clinton on 2000. But I was able to recognize the importance of nanomaterials. That's why I organized the International Symposium on nanoparticle and nanocomposite chemistry in the year of 1996, which is 4 years earlier than Clinton. So I'm very proud of having the kind of conference in Korea. Probably this is the first nano conference in the world. Yeah. Well, so my first paper as a Clay hybrid, which was on the DNA intercalated LDH, was appeared on the JACS, Journal of American Chemical Society, in 1999. And we extended this work to the gene therapy by intercalating antisense into the lead double hydroxide, which was published in Angewandte Chemie in 2000. Well, this kind of idea was extended not only to the gene therapy, but also to the chemotherapy and also radiotherapy by immobilizing the radioisotopes into the lead double hydroxide. And then currently we are actually applying this technology to the COVID-19 therapy. Probably, if I have enough time, I will show you shortly the current results of the COVID-19 therapy. So, as I, actually, we have published the DNA intercalate phase, as I told you already, and this paper was appeared in Angewandte Chemie as the cover paper. But later, this paper was pretty much highlighted by the Nature. The Nature journalist mentioned that the clay sandwich could smuggle DNA into cells using gene therapy. Yeah. Actually, this is the first attempt to apply the 2D clay for the gene therapy. And later, we have extended this research for not only for the in vitro, but also we applied this model for the animal model. And this was also selected as a cover paper in the Angewandte Chemie in 2016. But actually, this in vitro data was pretty much highlighted in CNN News and also the 2001 MRS meeting. And our research was selected as the one of the 8 innovative research, which was actually highlighted not only with the class label on the Tannenbaum and Peidong-Yang process. So, well, after publishing this nanobio compound in the JACS and Angewandte Chemie in 1999 and 2000, the MIT Technology Review announced that the 2 important nanobio hybrid technology, as you can see here. One is the nanomedicine, the other one is the nanobiomechanics out of 10 emerging technologies. So in the beginning of my talk, I will tell you what the nanoconvergence would be with a 2-dimensional solid like a clay. And I will explain you what the drug delivery system is and how we can realize the new drug delivery system based on the 2D nanomaterials as a carrier. And I'd like to show my synthetic strategy with you based on the molecular engineering and crystal engineering. And the rest of my talk, I'm gonna bring some experimental findings on the cytotoxicity of the anionic clay and interaction mechanism between clay and the cellular membrane and protein. And finally, the targeting function of inorganic clay and so on. Based on the basic understanding of 2D nanomaterials like clay, I'd like to demonstrate how we apply 2D materials for the drug delivery system, particularly for chemotherapy and for COVID-19 treatment as the proof of concept. And finally, I will summarize my talk with some conclusion. Then why DDS? There are several issues in the drug delivery community. For example, as you can see here, there are a lot of challenging things to do. For example, control release and solubility and bioavailability enhancement and taste mastery and overcoming drug resistance and the passive and active targeting, particularly for the chemotherapy. Well, of course, that kind of idea can be extended to the radiation therapy and also the tissue engineering. In order to apply the clay nanoparticle for the drug delivery system, we have to understand how the drug delivery vector could be functioned. For example, out of various structural materials, the liposome is the most intensively studied, the carrier. What is the liposome? Liposome is nothing but the molecular assembly of amplified molecules. Yeah. In the liposome, modified depending upon your design. For example, if you want to enhance the solubility, then polyethylene glycol could be conjugated on the surface. If you want to enhance the targeting function, then probably you can bring the antibody. And if you want to enhance the, for example, diagnostic function, probably you can probably bring some, dye molecules on the surface, or radioisotope on the surface, or something like that. Yeah. So that kind of drug delivery strategy is also valid for the other drug delivery things, such as polymeric nanoparticles, or polymeric micins, or dendrimer, and also inorganic particles like silicon nanoparticles, or the carbon nanotubes and porous carbon, and titanium oxide, whatever. But I'm very much interested in the 2D nanomaterials, as I mentioned earlier, particularly in the anionic clay, such as a hydrotalcite. Well, probably many of the colleagues here could understand what the crystal structure of lead hydroxide, but it is very important to understand the crystal structure from the brucite, magnesium. In the brucite structure, magnesium ions are octahedrally coordinated with the hydroxyl ligands to form an octahedron. And these octahedrons are bound one another by sharing the edges to form an infinite sheet. Yeah. And this infinite sheet are stacked on the top of each other to form a multi-layer. But interlayer, interlayer interaction is kind of very strong, bonding. So that's why it is impossible to immobilize the molecules into this two-dimensional lattice. However, if you, if the magnesium ions are partly substituted by trivalent cations, such as iron 3+ or aluminum 3+, or other trivalent cations, then positive layer charge can be generated. In order to compensate the positive layer charge, the anion, interlayer anion, can be stabilized in between this two-dimensional lattice. In such a way, the charge compensation could occur to get the thermodynamically very stable phase. However, depending upon the synthetic, strategy, one can immobilize not only the carbonate but also the nitrate, or chloride, halogenide, or acetate, whatever. Yeah. So in such a way, we can prepare the drug for gene clay hybrid by simply ion exchange reaction. For example, if you have a negative molecules, one can immobilize the organic molecules into this two-dimensional lattice by simple ion exchange reaction, or you can prepare that kind of hybrid by simple co-precipitation reaction in the presence of salts, the anion species, what you want. Well, in order to apply our nanovector for drug delivery system, it is required to check its cytotoxicity prior to its drug efficacy experiments in vitro or in vivo. And therefore, we have performed the MTT assay and tree-pumple assay in three-dimensional cell culture lines, such as L132 human normal lung cell, and Hep2 normal hepatoblastoma cell, and MCF7 is a human, human breast cancer cell with respect to the concentration. As you can see here, clearly that our drug delivery carrier is not toxic up to such a high concentration of around 500, 500 microgram per mil. We have also done some membrane damage based on the lactate dehydrogenase leakage assay. But we found that our nanoparticles did not make any significant damage on the cell membrane up to 100 microgram per mil. And in order to apply our drug delivery carrier for IV injection, we have studied the hemolysis effect of our drug delivery carrier, but no significant hemolysis effect could be observed up to 100 microgram per mil, indicating that the present, 2D nanoparticles, clay particles, are thought to be useful as a drug delivery carrier. Yeah, so we came up with an idea to understand the intercellular uptake mechanism of nanoparticles. And then what is the, we have to understand what is the endocytosis mechanism of our inorganic particles. And what is endocytosis? Endocytosis is a general term for the process whereby the large alien particles are wrapped with a plasma membrane and transfer into the cells in the form of a vesicle or vacuole. So, and now we are going to understand the endocytic mechanism of our anionic clay nanoparticles. According to the literature, there are multiple endocytic pathways in mammalian cells. As you can see in this figure, namely the macropinocytosis, clathrin-mediated endocytosis, caveolate-mediated endocytosis, and et cetera. But from the preliminary TEM study, the macropinocytosis could be excluded since the large reference could not be observed in the TEM studies. So we decide to check both the clathrin and the caveolate-mediated endocytosis. Well, to determine the specific endocytic pathway of nanoparticles, the immunofluorescence microscopy or confocal laser scanning microscopy are often employed. When the nanoparticles were treated in the cell culture lines, the membrane proteins are assembled there to wrap the inorganic particles around the inorganic particles, as you can see here. So we wanted to understand what sort of membrane proteins assemble there to, wrap the inorganic particles. So therefore, we have actually prepared the dye-conjugated LDH first. From the green fluorescence, one could understand where the inorganic particles are on the cellular membrane. So, but we wanted to know what sort of protein molecules are responsible for the formation of vesicle, clathrin, or caveolate. Since the corresponding antibody and the anti-antibody with the dye for clathrin and the caveolin were available. So cellular membrane was assayed by incubating the cells within antibody revealed by dye-conjugated secondary antibody on the permeation condition. So, from the co-localization of the green fluorescence and the red fluorescence, one could understand what sort of membrane proteins are assembled around there to wrap the our inorganic particles as a clathrin or caveolate. As you can see, the immunofluorescence microscope image here, both the clathrin staining fluorescence and the LDH green one overlap each other perfectly, as you can see here. But in case of caveolate, no clear image could be seen when treated with the LDH fluorescence as shown in the bottom of this figure. This result clearly suggests that clathrin-mediated endocytosis is the principal mechanism for the cellular internalization of the anionic clay, and cavially mediated endocyte. There's nothing to do with the LDH pathway. Well, after getting these results, we have discussed with the medical doctor, the oncologist at the Catholic Medical School and so, and they recommended us the anti-cancer drug methotrexate, as you can see here. And methotrexate has the function of carboxylic acid at the end of this molecule. So I thought that this molecule can be immobilized into the layered double hydrogen. You know, methotrexate is a folate, and this is used to treat a variety of cancers, including, you know, osteosarcoma, acute lymphoblastic leukemia, and breast cancer, and head and neck cancers by inhibiting the metabolism of a folate. However, MTX has been limited because of the drawback of the MTX. MTX has a very short plasma half-life and drug resistance, and therefore high dosage is always required for cancer cell suppression. In this study, you know, a drug delivery system is proposed to overcome such limitation. If MTX is internalized inside the cell, through the folate receptor, and then the MTX could be conjugated on the dihydrofolate reductase, which is involved in the. If this dihydrofolate reductase is deactivated by the conjugation of the MTX, then, you know, folate cycle will be stopped, and such a way cell proliferation and DNA synthesis and cell proliferation will be stopped. This is the action mechanism of this drug. So cell-based events. So, and now we decide to develop chemically well-defined MTX, you know, LDH nanohybrid drug delivery system to overcome such, you know, the drawback of MTX, such as a very short plasma half-life and drug resistance. Well, let me tell you how we prepare the colloidal MTX LDH nanohybrid structure. But first, LDH was prepared by the simple co-precipitation method, and thus prepared, you know, precipitate was further hydrothermally treated to obtain a colloidal drug by controlling the hydrothermal condition, such as reaction temperature and time. There is a very tricky technique there to get the colloid from the, you know, the precipitate. Yeah. It is very important to control the surface charge, yeah, upon treating the hydrothermal reactions. Well, according to the XRD patterns of MTX LDH, and the basal spacing of LDH was expanded from 7.69 angstrom to 21 angstrom upon intercalation of MTX into LDH layer. As you can see in the CM and TM images, the average particle size of MTX LDH was determined to be around 100 nanometer, which is in good agreement with the particle size distribution results obtained from Zeta Sizer. The cross-sectional high-resolution TM image in here, the interlayer distance between periodic lattice fringes from the 0, planes are approximately 21 angstrom, which is an excellent agreement with the excellent results here. So to compare the therapeutic efficacy of MTX and our clay drug, we performed the tumor growth inhibition test in the autotopic breast cancer model. The tumor volume were measured after intraperitoneal injection of the control solution, PBS, and drug carrier only, and drug only, and our clay drug into tumor-bearing mice. As you can see here clearly that the MTX anion clay group showed an even stronger anti-tumor effect than the intact MTX with a mean tumor volume of 600 cubic millimeter, representing approximately 81% and 74% reduction in tumor volume compared with the PBS control group and the MTX group, as you can see here. Well, here you can see the in vivo toxicity. The survival rate of MTX LDH is determined to be 100% compared to the control. Why? This result suggests that our clay drug significantly enhanced the therapeutic effect, and as a consequence, increased the survival rate compared to the free MTX, which is well consistent with the results of a biodistribution study, which will be discussed afterwards. According to our biodistribution study, we found that our nanoparticles are rather, accumulated in the beginning, accumulated in the liver in the beginning. So we had to track the liver toxicity. For tracking liver toxicity, in general, serum levels of AST, aspartate aminotransferase, and ALT, alanine aminotransferase, were studied carefully. And as you can see here in this figure in the left-hand side, AST and the ALT level of the LDH MTX injected mice were found to be in a normal range. Yeah. And we did also a histopathological study. According to the histopathological analysis of hematoxylin, AUC in the liver section, as you can see here in the right-hand side, does not show any morphological abnormalities related to the liver injury in any of the treatment groups. Actually, this analysis was made by the specialist histopathologist. And it is therefore concluded that our drug clay hybrid does not induce any liver toxicity and inflammatory immune response after the systemic delivery. Well, since the tumor to liver ratio is also a very important indicator of both the therapeutic efficacy and the safety profile, the enhanced tumor to liver ratio for MTX LDH compared to MTX only indicates its high potential as an effective and safe systemic chemotherapy agent. For that the tumor to liver ratio does not change with respect to time. However, in case of MTX treated group, tumor to liver ratio increases continuously with time. That our clay has the targeting function to the tumor. Well, now I'd like to shortly introduce the antiviral strategy of niclosamide LDH against COVID-19. And niclosamide is actually approved by FDA as anti-lymphatic drug. Oh, long time ago, maybe 1950 or something. Yeah, long time ago. But it was also known that the niclosamide has potent antiviral activity against the single-strand RNA virus, including various coronavirus families. And niclosamide was proposed as the antiviral drug during the severe acute respiratory syndrome outbreak in 2002. Yeah, and later it was found that this niclosamide could inhibit the SARS in vitro. Based on this, studies, many researchers hypothesized that the antiviral activity of niclosamide may be extended to the COVID-19. Yeah. Yeah. Unlike other antiviral drugs, niclosamide not only inhibits the replication of viruses, but also kills viruses. Yeah, as you can see here, the niclosamide has various antibacterial items based on the very side there, and also but recently, niclosamide could activate the autophagy. If the autophagy is, induced, the autophagosome containing virus could be fused into the lysosome, which is the cleaning compartment in the cell. In such a way, the virus can be, killed, destroyed there. Well, after the outbreak of COVID-19 3 years ago, a panel of 48 FDA-approved drugs that have been pre-selected by an assay of SARS-CoV and screened to identify potential antiviral drug candidates against SARS-CoV-2 infection. And it was found that 24 drugs exhibited antiviral efficacy against SARS-CoV-2. In particular, niclosamide exhibited notable antiviral activity, even against the mutant such as this mutant A or mutant B from England, B1.51 from the South Africa. So the drawback of niclosamide to limit its application of antiviral is the extremely low solubility and thereby very low bioavailability. We know that niclosamide has an efficacy in antiviral effect, but it was impossible to commercialize up to now because of its very, very low solubility. So in order to overcome such a disadvantage of niclosamide, such as very low solubility and bioavailability, we have developed a new drug delivery system based on the layered double hydroxide, namely hydrotarside as a carrier. Based on the synthetic scheme above there, LDH was first calcined at 300 degrees C. Actually, we have also performed the same experiment with the LDH only, but the solubility was not so well solved. That's why we actually calcined at 300 degrees C to induce the porous dihydrotarside, where niclosamide was coated on it to form a niclosamide DHT hybrid. Thus prepared niclosamide DHT hybrid was further formulated with the non-ionic polymers such as 2060 and the HPMC, hydroxypropyl methyl cellulose, for developing oral drug with a high solubility and bioavailability. According to the UV visible absorption analysis, as you can see in the right-hand side, we observe the characteristic P of pure niclosamide in ethanol, and this characteristic niclosamide P was retained in both NICDHT and after coating NICDHT coated with the 2060. As you can see here, without any specific peak shift could be observed in the absorption band, indicating that non-ionic polymer was coated on the surface of anionic anionic clay through the simple physical absorption that was not chemically modified on the surface. Here you can see the pharmacokinetic data, how the plasma concentration of nicosamide is retained with respect to the time. In the left-hand side, nicosamide plasma concentration at the 15 minutes of NIC DHT twin nanohybrid was around 1,350 nanogram per ml, which is higher than the highest nicosamide plasma concentration after oral administration of control drug umesine. Yeah. In the middle, the PK profile was subsequently optimized by changing the dose and the non-ionic polymer coating. The highest NIC concentration in plasma observed at the 15 minutes was significantly enhanced when the dose was increased from 50 to 200 milligram per kilogram. We found that the about 5-fold increase of the highest NIC nicosamide plasma concentration could be observed by dose increase. However, the time required to sustain the plasma concentration of NIC above IC50 remained at only 8 hours. So in the right-hand side, we attempted to further optimize the PK profile of oral formulation. And so NIC DHT surface was coated with the hydroxypropyl methylcellulose, which has a gastrointestinal, retentive property. The PK profile of NIC DHT HPMC drug shows that the nicosamide plasma concentration above IC50 was sustained until 24 hours. Yeah. And based on IC100 value, nicosamide plasma concentration lasts for 12 hours here. Yeah. Which is the champion data ever reported so far. And this graph shows the individual viral load in the control group and our drug treated groups on 2, 3, 4 DPIs, as you can see, 2, 3, 4 DPIs. Days, DPI means days post-infection. The standard model was the golden city enhancer infected with the SARS-CoV-2 with 105, PFU, which is the plug-forming unit through the nasal route. The viral copies in the blood was measured by RT-PCR. And the drug was continuously administered from 0 DPI and 1 DPI. And here, as you can see, the G1 group and G2, G3, G4, you can see here. And the G1 group was treated with our drug from 0 DPI, which means the immediate after infection. And in case of G2, G5 groups, were treated with our drug from 1 day after infection. Yeah, 1 DPI. The interval time for drug administration was 4 hours for the G1, G2, G3, G4, and 6 hours for the G5. As a result of experiment, low viral load than control group was observed in the G1, G2, G3, G4. Yeah. Low viral load was observed in the drug administration group than the control group. At the 4 DPI, overall viral load was upregulated, but the G4 group, which continuously administered 25 milligram per kilogram of our drug, show the low viral load. Yeah. So, we actually finished the preclinical study 1 year ago, and we have been doing the clinical trial 1, phase 1, and which was finished 2 months ago. And we are now on the clinical trial 2. And very recently, we have go through the, this clinical trial is divided into two parts. First one is the pilot survey, and second one was the pivot one. But the first pilot was successfully finished, and we are now on the way of second stage in the clinical trial too. I hope this will be finished very successfully in such a way. Probably we can see the first antiviral drug with the anionic clay. Yeah. Well, thank you very much for your attention. I'd like to conclude very shortly. And we found that our anionic clay are mainly internalized inside the cell through the clathrin-mediated endocytosis. And we also measure the particle size dependency on this mechanism. But we found that from the 50 to the 200 nanometer in size are internalized inside the cell through the clathrin-mediated endocytosis. If the particle size is bigger than 250, then no mechanism could be observed. And we are also very proud of preparing the injectable nanomedicine, MTX and MTX anionic clay. And also, we were very successful up to now. We were able to prepare the niclosamide LDH with HPMC coating as an oral formulation of COVID-19 treatment. But I think this is currently a very important social issue to bring our pandemic situation to the endemic. So we actually made a great effort to realize this niclosamide anionic clay drugs. Well, thank you very much for your kind attention.